Welcome to The Explainer. Today we're looking at one of the absolutely most incredible events in all of biology. But listen, I don't want you to just think of this as some dry, textbook scientific process. Really try to visualize this as a thrilling, high-stakes, microscopic journey. Millions are going to begin this race, but literally only one achieves the ultimate goal. We're going to break down the exact molecular steps, the chemical warfare, and the frankly mind-blowing cellular mechanics of human reproduction, focusing right in on the moment sperm meets egg. It's a microscopic gauntlet for sure, but it's also the very first chapter of your own origin story.
Okay, let's dive into this. How on earth does a single microscopic cell survive an absolute gauntlet and then breach a heavily fortified cellular fortress? You see, the egg isn't just sitting there unguarded. It's heavily protected, waiting at a very specific remote location called the ampullary isthmic junction deep inside the fallopian tube. So to really understand this epic breach, we first have to understand the defenses of the fortress itself. Right, so the egg boasts this brilliant double-layered defense system. The inner shield is the primary membrane, known as the zona pellucida. Now this is a tough, completely non-cellular layer made entirely of glycoproteins. Think of it like a hard shell. But outside of that is the secondary membrane, the corona radiata. You can picture this one like a thick, cellular crown made of 2 to 3 layers of granulosa cells, and they're all glued tightly together by a super dense ground substance called hyaluronic acid. Our winning sperm, it has to figure out how to get through both of these completely different types of barriers.
Now, hidden just beneath that tough zona pellucida and right above the egg's actual plasma membrane, which is called the oolemma, is a tiny but incredibly vital gap. This is the paravitelline space. I want you to keep this specific location in the back of your mind, because as you'll see a bit later in the explainer, this little gap, it's the ultimate battleground. This is the exact arena where extreme lockdown protocols are going to be deployed and where unneeded cellular materials will be forcefully expelled to ensure everything goes right.
And this brilliantly illustrates the sheer scale of the journey, because before a sperm even reaches those egg defenses, it has to survive a massive timeline of attrition. We are talking about hundreds of millions of sperm dropping down to just a few hundred survivors. Right at 0 hours, the sperm immediately faces the harsh, highly acidic pH of the vagina. If it manages to survive that, around hour 2, it gets trapped in dense cervical mucus or faces the complex maze of the uterine cavity. So many just lose their way entirely or get stuck in the ciliated epithelium and the fallopian tubes. Out of millions, barely a fraction reach that 6-hour mark at the ampulla. It's just a journey of extreme, unrelenting attrition.
But here's where it gets fascinating. The female reproductive tract doesn't just destroy the weak. It actively empowers the survivors through a crucial upgrade called capacitation. You see, the sperm actually arrive completely incapable of fertilizing the egg. They need a biochemical transformation. During this phase, the female tract naturally dissolves the cholesterol on the sperm's plasma membrane. And what that does is trigger a massive influx of calcium ions, transforming the sperm's slow, kind of undulating movement into this hyperactive, rapid whiplash action. It's a brilliant irony, really. The exact same environment that acts as a deadly obstacle course also fully weaponizes the survivors for the final breach.
So once our newly supercharged sperm finally reaches the egg, it encounters the ultimate biological security system. The egg's inner layer, that zona pellucida, has specific glycoprotein receptors called fertilizing. Meanwhile, the sperm is carrying an acidic protein called anti-fertilizing. When these two meet, it triggers an exclusive chemical handshake. It's a chemoattraction that acts exactly like a highly secure password. This is a strict, species-specific checkpoint, making absolutely sure that only human sperm can bind to the human egg's precise ZP3 receptors.
But, binding is really just the beginning. Now the sperm deploys its payload. There's a cap on the sperm's head called the acrosome, and it basically bursts open, unleashing a highly coordinated chemical arsenal. First up, an enzyme called hyaluronidase dissolves that sticky hyaluronic acid we mentioned earlier, literally melting the glue holding those outer cells together. Next, the corona-penetrating enzyme physically drills its way through the cellular layers of the corona radiata. And finally, acrosin goes to work directly digesting the hardened glycoproteins of the zona pellucida. It's just a perfectly orchestrated biochemical drill.
Let's move to this and see how this builds into a beautifully systematic breaching protocol. After that successful chemoattraction binds the sperm directly to those ZP3 receptors, that very contact is what triggers the acrosome burst, raining down those hydrolytic enzymes. Having successfully melted through both the cellular outer crown and the non-cellular hard shell, the sperm finally pushes its way entirely through to make physical contact with the oolemma, the egg's innermost plasma membrane.
And what happens next is honestly simply
beautiful. The egg doesn't just passively sit there and let the sperm break in. No, when that winning sperm touches the oolemma, the egg's membrane actually extends outward, forming what is called the fertilization cone. It literally reaches out, embraces the sperm head, and actively pulls it inside the cell. After all that, the successful sperm has finally arrived.
Now, what's really interesting about this slide is the sheer panic that ensues the microsecond that first sperm gets inside. The egg has to immediately prevent polyspermy, which is the fatal entry of multiple sperm. If more than one gets in, the whole thing fails. So it triggers an instant two-step lockdown. Step 1 is the fast block. A rapid influx of sodium ions instantly changes the electrical charge, depolarizing the membrane to literally shock away other sperm. But that only lasts for a brief moment. The permanent solution is the slow block, a massive wave of calcium influx that triggers the incredibly important cortical reaction.
This cortical reaction is the absolute, definitive sealing of the fortress doors. Deep inside the egg, these little sacs called cortical granules rush to the surface. Thanks to that massive calcium influx we just talked about, they fuse with the oolemma and dump their destructive enzymes right into that perivitelline space, that tiny gap we highlighted earlier. These enzymes instantly go to work destroying all remaining ZP3 receptors and drastically thickening the zona pellucida. Boom, the fortress is officially sealed. Any remaining sperm outside are completely locked out and will simply degenerate.
Now, while those doors are being heavily barred, there's actually some unfinished business inside the egg's nucleus. Up until this exact moment, the egg was actually frozen in a state of division, just waiting. The entry of the sperm definitively destroys a chemical called the metaphase-promoting factor, and that triggers the egg to finally, at long last, complete its cellular division, meiosis II. But it's an unequal division. The egg basically hoards almost all the cytoplasm to nourish the future embryo, essentially throwing the leftover genetic trash into a tiny secondary polar body, which is expelled right out into that same perivitelline space.
Okay, so now we have a fully mature haploid ovum and a sperm ready to finally combine. The contributions they each bring to the table are incredibly distinct, and they dictate the biology of literally every single person watching this right now. The mother provides the massive ooplasm packed with maternal mitochondria. These are going to provide 100% of the immense energy required for all the upcoming cellular divisions. The sperm, meanwhile, drops off its genetic payload, sure, but also a crucially important structural tool, the paternal centriole. You see, the egg lacks a centriole, so the father provides this vital little engine to organize the spindle fibers that will guide the very first cellular division of this new life.
So, the crucial point is, how do these distinct parts finally become one? Well, this elegant cellular dance is called syngamy. First up, in plasmagamy, the cytoplasm of the two cells merges together. Next, the nuclear envelopes completely dissolve during karyogamy, which allows those two distinct haploid nuclei to approach each other. Finally, amphimixis occurs, as the male and female chromosomes fully intermingle. The 23 chromosomes from the mother and the 23 from the father seamlessly pair up.
And at this exact incredible moment, we have a singular, diploid human zygote. The male sex chromosome, whether it's an X or a Y, has just determined the biological sex of the child. The epic, microscopic gauntlet is officially over. The chemical barriers have been breached, the fortress is sealed, and the genetic code is permanently set. But it really makes you wonder, how does this solitary microscopic cell now harness that maternal energy and paternal structure to multiply and construct a fully functioning human body? The journey of fertilization has ended, sure, but the absolute miracle of development, that has just begun. Think about that until next time.